Copyright disclaimer under Section 107 of the Copyright Act of 1976. Allowance is made for fair use for purposes such as research, criticism, comment, news reporting, teaching, and scholarship. Fair use is the use permitted by a copyright statute that might otherwise be infringing. Good evening, everybody. Hope everybody doing well. Like the show, share the show, tell a friend. We're live. I know I've been on a whole bunch today. This is what the third I think it's the third show I've done on this channel, and then I did one on the Mossy. What really pretty much wasn't a show, just pretty much a review from an astronomer's perspective, letting people see what the astronomers are saying. Doesn't sound like what the layman is saying, so we got to go and let the experts speak on said subject sometimes. Man, so I wanted to get into something that I figured would turn y'all heads. You know what I'm saying? And I'm gonna walk y'all through an actual study. This study is not going to refer to bat guts, but that's what it is. It's not going to refer to actual, lab grown bat guts. It is, but it's saying it in a different sense. Now, I'm gonna go ahead and tell y'all right now what they're looking for. What they're looking for is ways to be able to track disease, how bats carry diseases, and where they harbor the disease. Right? This is that real work. That real work. You know what I'm saying? Like from, a true perspective. You feel me? Like this is where that research is. So I don't know where everybody at, and it's cool. I don't. This channel doesn't garner a lot of attention, but for those of you who are in the building, man, like the show, share the show. I'm about to go in a little bit, and I really don't expect many people to hop in on this channel. You know what I'm saying? There was already somebody throwing shots and shade at those of us who actually talk our talk anyway. So I'm trying to let some more people come in before we get going, and we're gonna deal with it. I appreciate everybody tuning in. I was gonna do it. I actually would have been live right on time, but I got caught up in looking at some dudes competing on Instagram for like $10,000 or something like that, showing off their dogs. And the type of dogs that they had, it was some bullies. And this dude had a black bully micro. He was pretty cool. This other dude had a tri bully. He was dope. But ultimately, I like the try. But I don't know. That black one, he was just muscular. He was hard. He was hard looking. He was tough. I ain't gonna lie, he was tough. But we got something in store for that. We coming for them bags like that. We got something in store for that. But yeah, they cool. I'm gonna have to keep my eye on them, see how everybody vibing and rocking out. And then when it's our turn to unleash, we'll unleash. But please come on, man. Like the show, share the show, tell a friend. I don't know where everybody at, but we can get it in. And I also had some epidemiologist information that came in. But this was regarding some antiviral stuff. You heard the little beep come in. They just let you know that info just touched down. So I'm gonna go ahead and get started, man. People are just hold on. I'm gonna play the video first.
Hello and welcome to Med Simplified.
Hold
The
up.
immune system is the basic defense.
Hold up. That ain't the video that I want to play anyway. Let me go on and get y'all the video. I'm gonna play this video and then I'm gonna get into that science. I ain't even gonna lie to you. So just give me 1 second. 1 second. There go them commercials. I knew that was coming. Let the commercial play. It's gonna get real interesting. But I'm gonna play this video while we waiting on people to come in. This way, it help us out. Oh, my bad.
Awesome because they help us out in all sorts of ways.
Peep this.
One-fifth of all mammal species today are bats. And that's pretty awesome because they help us out in all sorts of ways. Like they pollinate a lot of plants, help regrow forests and control pests, and their poop is a pretty excellent fertilizer. Plus, they're just really cool. Some of them can sense magnetic fields or use sound to find their food. And of course, there's one thing that they all have in common. They can truly fly. They're the only mammals around capable of powered flight without the help of machines. But they're also somewhat notorious for something else, being flying sacks of germs. Now, you might have noticed that part with all the talk about zoonotic diseases that's been happening lately. Those are diseases that are passed to humans from other animals. And while bats aren't to blame for everything, they have played a role in the transmission of at least 11 viruses, probably 12, counting SARS-CoV-2. These diseases aren't the bats' fault, of course. If anything, they're ours. Research shows that disturbing animal habitats is usually what causes the transfer of a zoonotic disease to humans. Still, bats in particular do carry a lot of viruses, and that's because they have unique immune systems, which means we can learn a lot about these pathogens and their effects by studying bats. In fact, bat immune systems are so special that what we learn from them could someday help us treat a wide variety of conditions, from cancer to diabetes. And the kicker is, bats probably have weird immune systems because they fly. Unlike gliding, flapping flight requires a huge amount of energy. So bats have evolved ways to kick their cellular fuel production into high gear, mainly by putting their mitochondria into overdrive. Those are special compartments within cells that turn food into fuel. But there's a catch. When mitochondria convert nutrients into energy, they also create byproducts called reactive oxygen species. So basically, mitochondrial exhaust fumes in the form of really reactive molecules which contain oxygen. Now, these aren't all bad. The immune system uses them to rouse immune cells to action and kill bacterial invaders. But they can also cause a lot of damage. They can weaken cell membranes, mess with proteins, and even break DNA. And because of that, they play an important role in diseases like cancer and arthritis. Cells can try to keep them in check with antioxidants, compounds that essentially neutralize these overeager molecules. But those can only do so much. And when the balance gets out of whack, cells experience a condition called oxidative stress. This is when most of the DNA damage happens. So bats' supercharged mitochondria mean extremely high levels of oxidative stress, which in turn means constantly high levels of DNA damage. But since bats don't immediately get super cancer after their maiden flight, researchers have long suspected that they've evolved ways to protect themselves from all this flight-related damage. And a few years ago, genetic studies found mutations which boost their ability to detect and repair damaged DNA. Essentially, they've also turbocharged the mechanisms that prevent genetically damaged cells from replicating, which may also explain why they don't seem to get cancers very often. So bats produce tons of energy without damaging their cells. Sounds pretty awesome, really. But there's just one small problem. DNA damage can also be a sign of a viral infection, because viruses need to hijack the cell's genetic machinery to reproduce, and that process usually involves some strategic snippet. So naturally, DNA damage triggers an immune response, inflammation. Essentially, when cells detect DNA damage or other signs of infection, they chemically call in white blood cells. These cells kill and destroy pathogens using a variety of genetic and chemical tools. And they also help control how the inflammation unfolds, like by bringing in additional white blood cells or switching some of them from germ killing to tissue repair once the invasion is over. This immediate or acute inflammatory response helps to get rid of the invaders and promotes healing. But remember, thanks to their supercharged mitochondria, bat cells experience constant DNA damage. They can repair this damage thanks to those advanced DNA repair tools, but the damaged DNA should still light an immune flare before it's fixed. So bats would experience super inflammation all the time. And prolonged chronic and systemic inflammation isn't so great. White blood cells and the processes they set in motion can be really destructive to the body's own tissues. In short, too much inflammation can lead to organ failure and even death. So flight should be a death sentence. Except bats have evolved some neat ways to knock down inflammation too. For one thing, they dampen the activity of sting proteins. These proteins are one of the ways mammalian cells trigger an inflammatory response when a virus is detected. Also, a genetic analysis of multiple bat genomes showed that they're the only mammals that completely lack genes for pie-hin proteins, another set of inflammation triggering sensors activated by damaged DNA. And those are just part of the story. It will still be a while before we completely understand how bats prevent or dampen inflammation in their bodies, because it seems like every time they look, researchers keep finding more of these adaptations. So to recap, we know that to make sustained flight possible, bats have ramped up fuel production and DNA damage detection while dialing inflammation down to a 1. But we know that inflammation is one of the big ways the immune system fends off intruders. So doesn't that leave them open to all kinds of actual pathogens? And the answer is yes. Around the turn of the 21st century, scientists discovered that bats act as a reservoir for a lot of viruses that are extremely dangerous to humans. This includes filoviruses, which cause hemorrhagic fevers like Marburg or Ebola, and henipaviruses like Hendra and Nipah, both of which can cause fatal brain infections. And of course, coronaviruses like SARS, MERS, and most likely the notorious new coronavirus that started the COVID-19 epidemic. And there's mounting evidence that bats were involved in transmitting these diseases to humans, either directly or indirectly, like by infecting farm animals. And bats are also suspected of having given us other diseases in the past, like mumps, measles, and hepatitis B. But here comes another magic thing about bats. Even though they're widely infected with notoriously deadly viruses, they don't actually seem to get sick from them. The virus can be found in their bodies, but they don't have any symptoms. As for how that's possible, well, we still have more questions than answers, but researchers have discovered a few evolutionary quirks about bat immune systems that help make that happen. In part, that's because active viral infections in bats tend to be pretty short-lived, thanks to their hypervigilant interferon production systems. Remember how we said DNA damage is a signal of infection? That's because viruses try to reprogram cells to create copies of themselves. But cells aren't sitting ducks during this process. In addition to calling out for help, which is that whole inflammation bit we discussed, cells have an internal defense mechanism. They can make a protein called interferon alpha, which activates genetic and chemical tools that reduce the virus's ability to multiply and spread. Every other mammal we know of switches their interferon system on when an infection occurs. But genomic studies suggest bat cells always have their interferon.
All right, so the stream should be resuming in just a minute. I don't know why they try to block this video. This video could be played. They're trying to chase down $1 for it, I guess. Let me see if it pop back up. When it pop back up, it take a minute. Okay, it should be showing back up though. I'm waiting on us to pop back up on the live. Sometimes the YouTube just do this, but other times it just be that. What up, man? Okay, so it looked like I'm back now. And now that I'm back, you know what I'm saying? I'm gonna play it again. Sure.
And chemical tools that reduce the virus's ability to multiply and spread. Every other mammal we know of switches their interferon system on when an infection occurs. But genomic studies suggest bat cells always have their interferon alpha genes activated. This drastically cuts down the time it takes to react when a virus is present. So it allows bats to nip the infection in the bud before it becomes a full-blown disease. And of course, there's more. All mammalian cells contain an enzyme called ribonuclease L, which when activated chops up viral RNA to stop a pathogen from spreading. But in us and most other mammals, activating this enzyme takes a complex chain of steps, so it's not super quick. But bats, on the other hand, can activate it directly with interferons, drastically speeding up infection containment. Scientists also think this fast activation of ribonuclease L helps bats outsmart viruses that have evolved to inhibit the enzyme before it can be switched on, something HIV does in humans, for example. So basically, when bats do get viruses, they're able to quickly clear them out, for the most part. They can still fall prey to at least a few, like the rabies virus. And even quick suppression of a virus can mean it stays around in a group of bats. Bats tend to huddle close together when they roost. Plus, they fly around in the same areas as bats from other colonies, and they're constantly spraying snot and saliva everywhere when they echolocate. So there's a good chance that during that window, when a bat has a virus that is actively replicating, it will spread it to another bat. That means that even if each bat only hosts a virus for a short time, it can linger in the population. And even this probably isn't the full picture. Mathematical models indicate that by themselves, bats' social habits don't completely explain why they host so many viruses. Instead, research suggests that the viruses themselves have figured out how to lie dormant and undiscovered, like in the bat's lungs, spleens, or intestines. And then when the bat gets stressed, like when it's roused from hibernation, that stress temporarily dampens its antivirus systems, allowing any hidden viruses to emerge. This leads to another period of increased viral replication and shedding, so again, the bat can transfer the infection to other animals. Then its antiviral systems get back up to speed, and the viruses are eliminated or driven back into hiding. Still, even when a virus is replicating and being shed, bats don't generally seem sick, not like a person would with the same virus. And that's likely because their inflammation dampeners are still running. So they're still suppressing a lot of the immune responses that would make them noticeably sick. And that may also be why viruses that are deadly to us aren't lethal to them. It turns out that the most severe symptoms of illnesses like MERS and Ebola, the symptoms usually responsible for their lethality, aren't caused by what the virus itself does to the body. Instead, they're the result of the destructive, catastrophic inflammation the virus triggers. This includes an extreme systemic reaction called a cytokine storm, where an over-release of pro-inflammatory signaling proteins turns a person's own immune system into their worst enemy. And some researchers think that our aggravated inflammatory response may actually be because these viruses have evolved to dodge the super refined immune systems of bats. Basically, they evolved to survive in a host that constantly and aggressively attacks their ability to replicate. So when that assault is suddenly weaker in a new host, they go wild and produce a lot of little virus babies that send the...
I don't know if y'all caught what he said earlier about the whole thing with the bats releasing and things of that nature, because you couldn't see it. So we're gonna get into that, but I'm gonna let this last little minute squeak out, and then we're gonna get into
the host's immune system into panic mode. This kind of overreaction can also happen to bats. It's just not usually in response to viruses. Instead, it seems like their unique immune system may leave them vulnerable to non-viral invaders. The most infamous example of this is white nose syndrome, a fungal pathogen which has devastated bat populations in North America. Some researchers think the bat's dampening of inflammation, especially during hibernation, makes it easy for the fungus to infect the bat. Then, once the bat awakes, its immune system does react, only it goes too far. which can lead to the bat developing a life-threatening form of systemic inflammation. So basically, the reason they die from white nose is similar to why we die from viruses like MERS and Ebola. That may mean that the key to saving bats from this fungus and us from the deadly viruses they carry may lie in further research on bats. And also, if we're being selfish, we have a lot of other diseases characterized by inflammation, like heart disease and diabetes. So studying their inflammation system may lead to treatments for our chronic conditions. And the same goes for studies on the ways that bats keep viruses at bay. Right now, we only have effective antiviral drugs for about 10 of the more than 200 viruses that can infect humans, and very few broad-spectrum antivirals.
Y'all hear me? Oh, okay, cool. It looked like it cut off and then came back on, but it ain't showing on YouTube yet. This dude the worst. I know what it is too. All right, so we back. Let me get through this a little bit and then we'll actually I ain't even gonna play the rest of the little video. It's interesting what he was saying, but you know, we need to get into this study. That's really more important than anything. So let me share my screen. Look, they don't want to let me do that. So I can play him. We can hear him out the last couple of minutes. But he really made a hell of a point. He made a hell of a point back here somewhere. So I want y'all to hear this again, and then I'm gonna X him out.
When they roost. Plus, they fly around in the same areas as bats from other colonies, and they're constantly spraying snot and saliva everywhere when they echolocate. So there's a good chance that during that window, when a bat has a virus that is actively replicating, it will spread it to another bat. That means that even if each bat only hosts a virus for a short time, it can linger in the population. And even this probably isn't the full picture. Mathematical models indicate that by themselves, bats' social habits don't completely explain why they host so many viruses. Instead, research suggests that the viruses themselves have figured out how to lie dormant and undiscovered, like in the bat's lungs, spleens, or intestines. And then when the bat gets stressed, like when it's roused from hibernation, that stress temporarily dampens its antivirus systems, allowing any hidden viruses to emerge. This leads to another period of increased viral replication and shedding, so again, the bat can transfer the infection to other animals. Then its antiviral systems get back up to speed, and the viruses are eliminated or driven back into hiding. Still, even when a virus is replicating and being shed, bats don't generally seem sick, not like a person would with the same virus. And that's likely because their inflammation dampeners are still running. So they're still suppressing a lot of the immune responses that would make them noticeably sick. And that may also be why viruses that are deadly to us aren't lethal to them. It turns out that the most severe symptoms of illnesses like MERS and Ebola, the symptoms usually responsible for their lethality, aren't caused by what the virus itself does to the body. Instead, they're the result of the destructive, catastrophic inflammation the virus triggers. This includes an extreme systemic reaction called a cytokine storm, where an over-release of pro-inflammatory signaling proteins turns a person's own immune system into their worst enemy. And some researchers think that our aggravated inflammatory response may actually be because these viruses have evolved to dodge the super refined immune systems of bats. Basically, they evolved to survive in a host that constantly and aggressively attacks their ability to replicate. So when that assault is suddenly weaker in a new host, they go wild and produce a lot of little virus babies that send the host's immune system into panic mode. This kind of overreaction can also happen to bats. It's just not usually in response to viruses. Instead, it seems like their unique immune system may leave them vulnerable to non-viral invaders. The most infamous example of this is white nose syndrome, a fungal pathogen which has devastated bat populations in North America. Some researchers think the bat's dampening of inflammation, especially during hibernation, makes it easy for the fungus to infect the bat. Then, once the bat awakes, its immune system does react, only it goes too far, which can lead to the bat developing a life-threatening form of systemic inflammation. So basically, the reason they die from white nose is similar to why we die from viruses like MERS and Ebola. That may mean that the key to saving bats from this fungus, and us from the deadly viruses they carry, may lie in further research on bats. And also, if we're being selfish, we have a lot of other diseases characterized by inflammation, like heart disease and diabetes. So studying their inflammation system may lead to treatments for our chronic conditions. And the same goes for studies on the ways that bats keep viruses at bay. Right now, we only have effective antiviral drugs for about 10 of the more than 200 viruses that can infect humans, and very few broad-spectrum antivirals. If we can discover more of bats' tricks, we might be able to use them to develop therapies against the diseases they host and other dangerous viruses. Plus, all this research might help us live longer. Many researchers think bats' immunological adaptations are also behind some of their other superpowers, like how they seem to rarely get cancer, or how they live incredibly long lives for animals of their size. Usually, little animals live fast and die young, but Brandt's bats can live for over 40 years, even though they only weigh 4 to 8 grams. So instead of looking at bats as species zero, we should think of them as flying keys to longevity and resilience. And in the end, they're going to be our allies in health, not our enemies. And speaking of allies, before we go, we'd like to thank our patrons on Patreon. It takes a lot of people to make long, complex episodes like this come to life, and we can only bring all those people together because of the support of our patron community. So thank you. And if you want to learn more about joining this awesome group of people who helps keep SciShow...
And we ain't doing all that. All right, so here's your open access article regarding the establishment of intestinal organoid from Rosetta's leishmaniasis and susceptibility to bat-associated viruses, SARS-CoV-2 and piperine orthovirus. So there's a lot of authors associated with this particular article. Okay. Now this is an open access, and it's really only open access really due to the pandemic, but this was done a while ago, but people really wasn't paying it any attention. But we're going to pay it some attention. All right. Let me get big on the screen. Okay, that should help y'all out. But if you're on your cell phone, you know, you got to go and adjust your quality. You got to adjust your quality from 144 through the advanced and go on and make it 720. And as soon as you make it 720 on your cell phone, you will see it absolutely clearly. All right, so various pathogens such as Ebola, Marburg virus, Nipah virus, Hendra virus, SARS-CoV, MERS, CoV, and SARS-CoV-2 are threatening human health worldwide. The natural host of these pathogens are thought to be bats. The russet bat, the megabat, is thought to be a natural reservoir of filoviruses, including Ebola and Marburg viruses. Additionally, the russet bat showed a transient infection in the experimental inoculation of SARS-CoV-2. In the current study, we established and characterized intestinal organoids from a Leschot's rosette, Rosette's Leschotnaut T. The established organoid successfully recapitulated the characteristics of intestinal epithelial structure of morphology and appropriate supplements necessary for long-term stable culture were identified. The organoid shows susceptibility of uh, pterapine orthovirus, which is PRV, but not to the SARS-CoV-2 experimental inoculation. This is the first report of establishment of an expandable organoid culture system of Rousset bats. Uh, Intestinal organoids and its sensitivity to bad associated viruses and SARS-CoV-2. The organoid is a useful tool for the elucidation of tolerance mechanism of the emerging Rousset bat associated viruses such as Ebola and Marburg viruses. We can view the full text. We'll get into that in a second. Here we have an image of basically the bat, the intestines, and then the intestinal organoid. Okay. They look at the histology, and then through the microscope, here's the team, and then you have the marketer expression. B, you're looking at pretty much bat guts going through. C, intestinal tissue, and then D. So the next image, all right, intestinal organoids, intestinal tissue. All right. And then we have figure 3. So these are I need to show you these because they're getting ready to explain these images to us. Look at that, the ACE2. Y'all already know what's trying to go down in all of that. All right, so it's a lot of images and stuff like that. So look, let's just say we don't understand none of that. Cool. All right, now Google Scholar, you in the chat, your name right here. Let's go to the full text. Do I want to bore y'all to death? Yep. Mm, 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 mm. We can download the PDF if we want to, and then we really can break down the emphasis of the article, but we ain't got to do all of that. So here's the introduction of the article, but we kind of read the abstract already, so we know in which direction we're going here. But there was something very interesting in the introduction that I thought, and it's right here in this particular paragraph. And let me give it to y'all so you can see it. All right, it says, and I'm gonna have to, I think I got the whole thing on there now. All right, so it says, let me scroll up a little bit so y'all can see it. It says knowledge of bat specific immune response against virus infection at the organ level has been restricted by the limitations of the in vitro system. Three-dimensional culture systems such as organoids have been established and used to study disease modeling, drug testing, stem cell behavior, and epithelial responses to injury and host pathogen interactions in humans and mice. Let me continue. The intestinal tract is known to be one of the gateways for pathogens to enter and exit the body, along with the lungs. An expression of angiotensin converting enzyme 2 or ACE receptor 2, the receptor of SARS-CoV and SARS-CoV-2, was identified within the intestinal tissue with the same level as lung in Egyptian Rousset bat. Now, you heard me say this correctly. The intestinal tract is known to be one of the gateways for pathogens to enter and exit the body. Remember, we are specifically speaking about these bats. Now, in bat intestinal organoids from the Rhinolaphu sinicus, a natural reservoir of SARS-CoV-2 were established, and not COVID-2, I'm sorry, SARS-CoV were established, and it was shown that the organoids recapitulated the bat intestinal epithelium. These horseshoe bats, which is what you've seen um, from West Africa jumping up and down about in the chat, the horseshoe bat Enteroids express ACE2 and transmembrane cellular protist serine 2. The bat enteroids developed a cytopathic effect. An increased viral load was observed until 72H post-infection. However, the investigation could not consistently sustain long-term expansion as in human enteroids using the medium cocktail. Novel in vitro experimental systems are necessary to analyze virus-host interactions in fruit bats against Ebola and Marburg viruses and to isolate novel viruses from fruit bats. Therefore, we aim to establish and characterize intestinal organoids for the R. leishmaniae, assesses long-term culture methods, and evaluate their usefulness for studying host virus interactions. The results in the Rousset bat intestinal organoids were successfully generated and analyzed from the, which is the Rousset, is what they're dealing with, that died of natural causes at the zoo and were collected in Figure 1A. The structure of the organoid mimicked the multicellular structure of bat small intestinal epithelium, and the organoids recapitulated histology of the original tissue. In TIM analysis, the ultrastructural morphology of organoids mimic that of intestinal tissues. The absorptive epithelial cells with characteristics of microvilli, goblet cells, and Paneth cells were identified in organoids. All right, so this again shows you where they started with the bat, the intestines, then the intestinal organoids, put it under the histography under a microscope, market the expression, and they come up with the latter, which is those images right there. Now, to characterize the Rousset bat intestinal organoids, the expression of several specific cellular markers of intestinal tissues was evaluated by immunofluorescent staining. This is how we get the blue and the green against the black. Now, the ecadherin and epithelial cell marker CK20, a mature inner-site marker, and the MUC2, an intestinal globate cell marker, were detected in the organoids of the bat intestinal tissue, and both ecadherin and CK20 were positive only in the outer layer of the organoids, whereas the LGR5, an intestinal stem cell marker, was more abundant in the organoids than in the original tissue. These data indicated that the organoids recapitulated the cellular components of intestinal tissues with high steamuses. I'm not going to bore y'all to death. All right. So this is, again, we read the abstract and we know where we're going with this. We know we're in the results phase, but here's the discussion point. So they're looking for, small story short, they're looking for the harboring of these potential, like I told you before, these diseases, these viruses. I said diseases, but I need to correct myself. And let's just say the viruses. It's not disease. They're looking at these bats again, right? Their stomachs for places where they can harbor it. Now, remember, if you know anything about bat origins and so on, let me see if I can find some on you. Find some for y'all real quick. Let's see. Let's see if I can get it. I don't want to do this because we already looked at that one. But we could look at the pandemic. Nah, I ain't gonna be able to find it here, but there's a documentary that I think that is on Netflix that you actually can see where they go in caves and they look at the teeth. They look at the teeth on the bats to tell if they, how long the bat's been around. But they always did that, and then they would go in and try to, keep those bats and look at them and analyze them and so on and so forth. However, in this instance, the indication now is, where is the reservoir? Where are they harboring these viruses? So they found out by looking at the intestinals of a bat is a good place because they're in the gut. So we've identified I was about to say reservoir, but we identified a place where these viruses harbor, right? So because these bats are in unique places, we know that they're harboring the viruses in their stomachs. So if they release them through poop or whatever, yeah, really through poop, they begin now to be exposed. So I want y'all to think about that. Like going in a cave, and why they always say that you got to wear certain suits when you go into a cave, because you may be susceptible to potential pathogens you may never encounter before because of bat poop. So the science continues to back up that statement, really. On to some more good stuff. All right, so again, this has to deal with the gut as well. The microbiota of bats is a mutagenic properties and possible frontiers in preventing emerging diseases. So again, this is letting you know, again, this is open access to scientific reports in the Journal of Nature. And this is showing and proving how these people are looking for potential natural reservoirs for emerging viruses. That's the onset, but I'm not going to read the abstract to you. There's something that I want to get into uh directly that I wanted to pull up out of here, but let me make sure that I can hold on. Let me check where did I have it at? All right, I just scrolled past it. My bad. What a thing got to say intro, results, abstract, multi, discussion, methods, intro. All right, so I wanna pay attention right here in this article, right here, this part. It's the only thing I really want to deal with. I might get into this first paragraph here, but over the past years, bats have been widely studied as primary reservoirs for emerging deadly human viruses. There's a direct evidence that Nipah and Hendra viruses, which led to outbreaks in Southeast Asia and Australia, originated from bats. The evidence of bat origins of Ebola viruses is debatable, as it is based only on serological studies and detections of Ebola-related viruses in these animals. A similar situation with SARS-CoV and MERV-CoV, and especially SARS-CoV-2, and there is no unequivocal worldwide consensus on the origin of these viruses. However, the involvement of bats in the interspecies transmission of emerging viruses is undeniable, as evidenced by discovery of probable virus precursors of the above mentioned coronaviruses in them. Future epidemic and pandemic outbreaks of emerging viral diseases are inevitable. Even before the COVID-19, several papers were published where the possibility of occurrence of emerging zoonotic bat viruses have been discovered. Therefore, one of the main priorities today is to find ways to prevent or delay the spillover of highly virulent viruses from animals to humans. Bats should be the primary target in such studies, as the range of zoonotic pathogens in them is the highest among other known mammalian species. Also, these animals pose an additional threat of reservoirs of emerging zoonotic infections, as they are the only mammalians with the ability to fly and relatively long lifespan. This result in a high rate of contact with humans and, mostly important, other animals. The secret of viral biodiversity in bats lies in the immune and inflammatory response to infections. The central concept between this unique feature and the relationship between the ability to fly and resistance to viruses mediated by high levels of free radicals produced during the flight and subsequent DNA damage. In addition, some suggest that the gut microbiota of bats also could be involved in tolerance to viruses. However, The exact mechanism have been revealed. So in this study, it basically talks about what they're looking at, especially in the gut. So that's where we're at. This was published in October though, but it's mighty interesting that, what I'm saying, that they're looking at this gut like this. Let's see what show up if I do something stupid like that. See, I can play Science Inside and get away with it. But these people, because they do all of that old monetizing and selling stuff all on their stuff, it's difficult. I'm trying to find this, if I can find this documentary. Look at that. Oh, look, we on here. We ain't got that many people watching. That's a true bet right there. Wow. What's going on, y'all? Hey, man, that noise ain't in the chat, is it?
And the investigation into the origins of the coronavirus has taken a turn south, literally. A new U.S. government report has found that bats found in Laos, a tiny landlocked country which borders China, could be the basis of the gain of function research suspected to have been conducted by the now infamous Last September, scientists from the Pasteur Institute in Paris and the National University of Laos found 3 viruses found specifically in this particular bat species that were about 97% similar to SARS-CoV-2. It's believed these bat samples were sent to the Wuhan lab 1,000 miles away in China, where they may have been manipulated. So joining the conversation, our friend...
I did that on purpose, but look, this is because this all hinges on United States government documents, bro. When you looked at the title of this, you know what I'm saying? Like, I'm finna, but you know what documents they talking about, right? This virus that we're talking, these bats that we're referring to based on their criterion is just crazy. All right, so this is the document that they're referring to. If you've never seen this before, now you see it. There's no scientific studies in this that's going to tell you or me, y'all looking at this. Look, evidence of a lab leak. You or me, that it came out of a lab. There's no evidence in this 84-page document from the Republicans, from the U.S. government, the what they call itself, the GOP. From the members of the GOP, there's no scientific evidence that reports. Now look, they show all the hospitals. They're showing all this old stuff where people have activities. And you can go through every page of this, and you're not going to find any scientific evidence that supports a lab leak. And I'm not calling it a hypothesis or none of that, because it's neither. And then in each part of that, they give you conclusions like this. While much of the public debate was initially focused on the Hunan seafood market in Wuhan as the origin of the pandemic, the preponderance of evidence now suggests that the virus leaked from the Wuhan Institute of Virology. And if we go back and we look in the pages that we are dealing with and we read it, for instance, right? Look at what they say here on this page that I just left. It says research in France also retested samples from late 2019 in an effort to identify the COVID-19 cases. They identified a 42-year-old male who presented in the emergency room on December 27 with influenza-like illnesses. He had no connection to the PRC and no recent travel history. That has nothing to do with anything. You're looking for the origins, bro. So in here, we should be talking about the lab. We should be talking about the lab and how you found DNA evidence in the lab. There's no DNA evidence or no lineages. Am I saying this? I'm saying this wrong. There's no evidence coming out of the lab that says that it was there and that's where it started. Nothing is leaving the lab. Everything is coming to the direction of the lab and beyond it, from outside of near any lab. But this is what we're looking for. Evidence, right? We calling this evidence. This is he say, she say. It says the evidence above, combined with a cover-up conducted by CCP authorities, strongly suggest the Wuhan Institute of Virology as the source. So they suggest it, but they don't show any evidence. And then look, it says evidence of genetic modification. Watch this. The other topic to debate is whether the virus could have been genetically modified. The Wuhan Institute of Virology was conducting gain-of-function research on coronaviruses and testing them against human immune systems and in the months leading up to the emergence of SARS-CoV-2. However, the scientific community has claimed it is not possible. It was anything but a natural occurring virus. So the scientific community claims that the virus is natural. But as this report lays out, we believe it is a viable, look, y'all, a viable hypothesis. The scientific community says that this is a naturally occurring virus, but the GOP says this is a viable hypothesis that the virus could have been modified. And then they have a quote from Ralph Baric. You can engineer a virus without leaving any trace. Smack this Negro. Smack whoever he smacking. So we go on. And you can look at the bottom of each page where they have these sources. None of this stuff has anything to do with scientific data. This looks like it has something to do with scientific data until you get into it and see it has nothing to do with what they're trying to prove their argument for. This is only suggesting what France did when it came to their origins. So now we got the bat lady, right? So they're going to try to use the bat lady. It says research regarding SARS-like coronaviruses from 2004 to 2017. There's nothing in here. Look, beginning in 2005 and continuing 16 years, she and Desiree collaborated coronavirus research. Okay. Then it says they have identified more than 500 novel coronaviruses, including roughly 50 related SARS and MERS, and they have repeatedly engaged in gain-of-function research coronaviruses designed to make them more infectious in humans. So it says source number 80 for that, right? So we should be able to look a bid, which means this is the source. So they're using in Wuhan with Batwoman at the origins of COVID-19. They're using this to say something. But do the Batwoman really say that? Let's go on. It said it was also funded by the U.S. government. And then they go through and give some funding information. Purpose: the scientists hope to identify the origin of SARS by identifying species of bats which are natural hosts for SARS-like coronaviruses. Conclusion: these findings of coronaviruses, together with data of heniopapavirus, suggest that the genetic diversity exists among the zoonotic viruses in bats, increasing the possibility of variants crossing the species barrier and causing outbreaks of disease in human populations. It is therefore essential that we enhance our knowledge and understanding of reservoir host distribution, animal and human and animal interaction, particularly with the wet market system, the genetic diversity of bat-borne viruses to prevent future outbreaks. Relevance: this conclusion will drive the next 15 years of collaboration between the Wuhan Institute and Peter Daszak, and she directing the laboratory. Look, 2006, 2007, like none of this stuff has anything to do with any science. I'm telling y'all, they put together anything for argument. Anything. One of these chimeric viruses was able to enter cells through the human ACE2 receptor. The ACE2 receptor is abbreviation for the antigen tension converting enzyme 2, which is a protein found on the surface cells throughout the human body. You don't. They playing politics with no scientific information. We won't find one study. Look, watch this. Article publication: isolation and characterization of a bat SARS-like coronavirus that uses the ACE2 receptor. They use this source, right? They use this source to try to make the ACE2 receptor binding argument here. We all know about the ACE2 receptor. We've been paying attention to the pandemic, but that does not say that this has anything to do with gain of function. Because if you're gonna deal with the ACE2, we need to see this. We need to see what they're saying, and we know right now you ain't saying this. I done read this thing like 4 times trying to look for information. Researchers concluded that a minimum insert region is sufficient to cover the SARS-CoV-2 SARS-like coronavirus spike protein from non-ACE2 binding to human ACE2 binding. They're not using anything to prove it. Why they not talking about the receptor binding domain? We have evidence about we have once they map the genome, bro, you can go back in there and look and tell what you need to tell about the virus. You get under a microscope. There's no laboratory work done in this particular argument. This is merely a political onslaught of wasted time. What page is this? 33. There's no scientific studies in here. None. Look what this says. This paper is significant because the author discussed moving from from disease surveillance to creating chimeric viruses as a means of pandemic preparedness. This manuscript describes efforts to extend surveillance beyond sequence analysis, constructing chimeric and full-length zoonotic coronaviruses to be evaluated. During this work, researchers produced chimeric viruses created by inserting a spike protein from WIV1 into a strain of SARS-CoV, adapted to infecting mice. They subsequently tested this chimeric virus in human airway epithelial cells as well as in mice. In addition to standard Bal C mice, a strain of albino lab-bred house mice used in experimentation. Researchers genetically manipulated the mice to create a strand of the mice expressing the human ACE2 receptor. While the ACE2 found primarily in the lungs of the mice, it was also present in the brain, liver, kidneys, and gastrointestinal tract. The WIV1 chimeric viruses has been tested in these ACE2 expressing mice, proving that the chimeric, this didn't prove it, but proving that the chimeric virus could infect humans. So they used something that had to genetically modify the mice, right? Y'all remember this. I told y'all this a long time ago. They genetically modify the mice so that they can be infected with a virus. But in this instance, they're trying to use a different virus to show something in the study, right? And what they're looking for is to see if it'll infect the mice. Well, you genetically modified it so that it would infect the mice, duh, but you didn't do this. There's no experimentation or lab work to show that this same thing that you just did to the mice and you introduced it to a human, it infected a human. You just came out and said that this could infect humans. That's what I mean by there's nothing. It's always maybe, could, possibly. This whole 84 pages is about maybe. It could, possibly. It's all doubt. This whole paper is driven by doubt. And nobody would actually understand that if they just, if they just looked at like, oh, the Republicans. No, man. Have you read it? Read it. It's full of doubt. Watch. I'm going to go to all the conclusions. I'm going to go to the next conclusion. I'm going to read without reading anything. I'm going to read y'all the next conclusion. And you tell me what you think of the conclusion. Now, these are senators and what's the other ones? You got the Senate and then you got the House. State representatives, whatever they call themselves. Just watch. I'm going to get to it. They got old 2005 stuff. They ain't got nothing. They talking about some 2005. Ain't got nothing to do with 2020 or 2019. Wait till I get to it. I ain't got that yet. Where is this conclusion at? Look at that. Evidence of a lab leak cover-up. Now they covering up a lab leak. Look, they talking about April 2012. 2016. Look, there, boy, they try hard. We found that a short region of RNA-dependent RNA polymers from bat coronavirus, which was previously detected in Rhinolophus affines of Hunan province, showed high sequence identity to 2019 in COV. We carried out a full-length sequencing. Y'all didn't do that. You're a damn lie. Finally, the receptor binding domains show up in this article on like the 40-something page. But they talking about something 2015. And it's going to come back to doubt again. And I wish I had a team of researchers, like 20 people that all did their research on it. Then we just came back and did something that like they be doing. Cover up. You don't stop. But when they talked about that money, boy, it was funny. That money was funny to me. All this is to try to really come and vouch it. Look, I can't make this up. I can't make this up. They didn't want to Twitter on y'all. They didn't put Twitter clips in here. There's still no conclusion like it was in the earlier onset, by the way. I'm looking for it. I want to read it to y'all. But look what the GOP says right here. However, misinformation, unsubstantiated claims, and personal attacks on scientists surrounding the different theories of how the virus emerged are unacceptable. But why y'all doing it then? And are so in public confusion and risking undermining the public's trust in science and scientists, including that. Like, you know what I'm saying? They with the pump fake. They put that in there to try to, I don't trust scientists either, man. I want to see the evidence too. I want to see that. I want to see your evidence. This we ain't got a lick of evidence, and we ain't even came to a conclusion yet. All right, let me get out of this. Let me back. I'm on 58. All right. Let's have some fun. The intelligence community. That should be better. The intelligence community 90-day review report on the origins of COVID-19, ordered by President Biden, is due no later than August 24th, 2021. While based on open source information, it is the hope of the committee, minority staff, and the collection of analysis contained within this addendum produced by the direction of the ranking member, Michael T. McCall, will help inform the public debate about the viability of the laboratory accident being the source of SARS-CoV-2. It is vital the public discourse around the Wuhan Institute of Virology is transparent, honest, and detailed. So, what does Michael McCall say in his conclusion? It is the opinion of the committee minority staff. It is the opinion of the committee minority staff, based on the preponderance of available information, the documented efforts to obfuscate, hide, and destroy evidence, and the lack of physical evidence to contrary, that SARS-CoV-2 was accidentally released from a Wuhan Institute of Virology laboratory sometime prior to September 12, 2019. The virus, which may be natural in origin or the result of genetic manipulation, was likely collected and identified in the Hunan province, PRC, sometime between listen to this, y'all, look, between 2012 and 2015. They're going to tell us that a novel virus that emerges in 2019 likely was collected and identified in a cave in Hunan province sometime in 2012 and 2015. Its release was due to poor lab safety standards and practices, exacerbated by dangerous gain-of-function research being conducted in inadequate biosafety levels, including the BSL-2. The virus was then spread throughout the central Wuhan, likely via Wuhan Metro, in the weeks prior to the Military World Games. Those games became an international vector for spreading the virus to multiple continents around the world. Did y'all hear what I just said? They said that the Wuhan Military World Games is how the virus got out and it spread to everybody else. Come on, man, hit the like button. Don't play with me. This is your GOP member, Michael McCall. This is his team. This is the GOP study. This is everything that they claim that they confiscated out and they put into an 84-page document. And this is what he came out with. It is the opinion of the committee minority staff. So this is an opinion. And then you get a novel virus that they collected in 2012, 2015, and then released it. And then they released it during the Military World Games. I can't make this up. Nah, not me. I can't make it up. Then he goes on to say, it is the incumbent on all the parties identified in this report responded to the issues raised herein and provided clarity in any new or additional evidence as soon as possible. As always, committee minority staff stands ready to receive such evidence or testimony that supports or contradicts this report. Until such time as the Chinese Communist Party lifts its self-imposed veil of secrecy, explains its lies regarding the early stages of the pandemic, and provides access to the WIV's archives and the sample database, questions will remain as the origin of SARS-CoV-2 and COVID-19 pandemic. Until that day, it is an incumbent upon the United States and like-minded countries around the world to ensure accountability and implement the reforms necessary to prevent the CCP's malfeasance from giving rise to a third pandemic during the 21st century. Now he goes in with this timeline. Timeline of Wuhan Institute right here. This is the timeline, y'all. This is the lab leak and the start of COVID-19 pandemic. April 2012. According to the GOP, April 2012, when the 6 miner workers in the copper mine located in the Cable province of PRC fall ill. 2012 to 2015, researchers from the WIV collected samples from the bats in the cave. 2015 to 2017, Xi Jinping and all the other Chinese and Peter Daszak jointly published research on the isolation of novel coronavirus. They conducted the gain-of-function research testing novel and genetically manipulated coronavirus against mice. When was the first SARS breakout? Does anybody know when SARS-1 hit the scene? Because I believe SARS-CoV was first detected in those 6 gentlemen, but this was SARS-CoV. It was the regular SARS-CoV. But that outbreak occurred, the first SARS-CoV, I think, was first identified in 2003. And it only spread to 4 other countries. If I'm wrong, somebody can correct me on that. So let me try to figure out what's up with this 2012 SARS-CoV miners. Somebody can help me out with the regular SARS. That'll be nice. But if not, then that's cool. Ain't no thing but a chicken wing. So the claim is that by the GOP now, the claim is that COVID-19 first appeared in the Chinese miners in 2012. That is the claim that's being made here by the GOP. And this was their position. And then they go on and then they say all this other stuff. They talk about the mice, they talk about all this other stuff, and then they skip all of it from 2015 to 2017, then they go directly to 2018, 2019, and they're still talking about mice. And then they say they got an unpublished novel and genetically modified coronavirus. They're pointing the hell out of the finger at them. And then they come through and they say all of these other things. And then as we get closer to the pandemic, it's around the world. And then they continue. I can't make this up. It's unfortunate. This information is way out of whack. And you get mad real easy. Look, to Peter Daszak from Ralph Barrick, and they blocking out names. No need for you to sign the statement, Ralph. I spoke with Linfa last night about the statement we sent around. He thinks and agree with him that you, me, and him should not sign the statement, so it has some distance from us and therefore it doesn't work in the counterproductive way. These are the things that they were trying to get out of the emails, right? So this is one of their plays about Fauci, is that they were trying to get into the emails to try to demonize him or link him to communications that would suggest that he used governmental funding to support gain of function during that time period, uh, and it was just whacked out. So this whole, you know, this whole thing associated with like the bats and the bat guts and all of that is significant. Oh, November 2002. Okay, cool. 2002, 2003. We'll take that. We'll take 2002, 2003. We'll see. We'll just say it like that. 02, 03. My thing is that there's no way that in 2012, when these people fell ill, that they began to create a virus that'll be released in 2019 at a military event. Because, all right, so here's why I'm saying what I'm saying. Because the virus would have escaped the lab before before the end of 2015, 2016, 2017, 2018, and 2019. Literally 4 years. I mean, 7 years. They have, based on your report, 7 years they have been manipulating and trying to create this particular. It would have escaped because it would have infected one of those people. It seriously would have infected those people. They would have came out of that lab sick as a dog, and it would have already. It would have been. It wouldn't have been COVID-19. It would have been COVID-16. This report totally ignored the WHO. They actually condemned the WHO. They said that the WHO and the Chinese and the Wuhan Institute was working. Wuhan Institute of Virology was working together. The WHO did an independent study, and the GOP comes to the conclusion that they were partners. So what y'all think about what I just said? Or what y'all think about them studying bat guts? What y'all think? Nobody got, y'all don't know how to think now? Nobody got nothing to say? I'm waiting on y'all, man. I know it's a delay. Black Dot Bookstore, 12222. Chill Her, MC Chill Her YouTube channel, 1722. Next Friday, 7 p.m. Eastern. I'm on Chill Her's YouTube channel with Dr. Cole. We going in. Hopefully she will introduce some important information to us. Also, 12222 Black Dot Bookstore, Atlanta, Georgia. It's going down. Free event and book signing. You're going to hear from several different gentlemen regarding the pandemic. We're going to get into some tough questions, and we're going to handle our business like G's. What you mean you wish you would have known? We just told you before. Change it. You got time. But it's all good. Don't change it. Listen, y'all. Everybody keep sharing this dude post, bro. Like, listen, the dude need attention, okay? The dude need attention, and he's looking for people to say something to him. He needs attention. He's going to continue to look for it until somebody gives it to him. Let the dude die. Who cares? Let him do what he do. You know what I mean? We dealing with real world solutions, bro. And if you want to go gossip and do all the other stuff, then you do it. Let him do him. The blind lead the blind all the time. So shit, let him do his leading. I guess I don't know. I wish I could be there. I told a few about it who live in Lithonia. Okay. Word. But we're gonna handle our business and we're gonna do it like G's. So I just need all y'all in the building, man. If you're in the Atlanta area or around the Atlanta area, if you're outside of Georgia and you're only a couple of hours away, man, pull up on us at the Black Dot Bookstore on 12222. I'm 3 hours away. So if I can drive 3 hours, you know what I'm saying, you can ease on down the road too. Let's make it happen. It's gonna be a book sign. There's gonna be plenty of books there. Kofi gonna be there. You know what I'm saying? Nahisi gonna be in the building. Panther won't be there. Sutek on the fence. Chavez, he might be there. You know what I'm saying? I don't know yet. He might bag out. You know how black people get. So we going up and we gonna handle our business. I'm telling you. We're gonna handle our business. I'm surprised ain't nobody get. Hey, but look, who wanna argue? Go get them. Let's all let there go the link. They've been talking all that stuff on your social media pages. Tell them come join you. Hit the link, man. That's all I got for you. Hit the link. Let me hear what you got to say. Educate me. What I'm biased talking about? I'm missing. Let me go find out what I'm biased talk about on this other page. I don't think it's subscribed. I'm biased over here lying, bro. I'm biased got 12,000 subscribers, yo. Am I on the right unbiased channel? No, this ain't the right one. What's his other channel? This dude got so many channels, man. Well, he ain't live no more. What did it do? Go off? I can't see it no more. There you go. He called it unbiased talk. I couldn't find it.
Okay, then. Shut the fuck up. They look better than yours.
And so show improve, bitch.
All black people got jungle hair, yeah.
Show improve,
We definitely show improve. But
Here we go.
the difference is most of the people on this motherfucker have already seen me. I don't gotta show improve shit, bitch.
I know.
That's the difference. You're the Johnny come lately. You're the Johnny come lately, honey. You're the one that just got here. I've been here, honey.
I am the one who
Look who all in their chat. Look who all in their chat. Pure Black in their chat. Onk in their chat. Onk in their chat. Pure Black in their chat.
And that means so much.
Zane in the chat.
That means a lot.
Zane in the chat.
That means a lot. That means a lot to you.
You know what I'm saying?
You don't even realize that.
You see what I'm saying?
So you don't even realize that, bitch, you were invited as the entertainment. You're the jester, honey. You're the entertainment, bitch.
And that is who is that talking like that? Look at him. Look at look at him. Look, Pure Black. I'm up here. He said I'm here too. Look at him over there listening to them call each other bitches. Hey man, this is what we come. Hold up. Can't nobody say it like brother, man. Hold up. Let me. If somebody go get some credit for my views, it's gonna be my boy. You know what I'm saying? It's gonna be my boy, man. Hold up. Can't nobody say it like my boy, man. I don't hear no haters.
Tell the truth about me. Tell the truth about me and apologize, and I'm gone. I don't want one done. Apologize, and I'm gone. And here's the other stipulation. Do it within 7 days. Do it within 7 days. Within 7 days, tell the truth and apologize, and I'm gone. If I gotta keep drawing this out, if I gotta keep stepping away from my motherfucking bread, I gotta keep stepping away from my $12 million fundraise. My $12 million fundraise. If I gotta keep stepping away from my desk to answer these phone calls, just do it within 7 days. Apologize, and I'm gone.
This is what it come to. This is what it come to. This is what it come to.
I guess this is what it come to. Yeah. Hey man, this is what it come to. Tika said if a person gets SARS-CoV-2 right now, how would the doctors know which strand they have? Well, first let's clean that up. When they do PCR testing, they can sequence the genome to determine which variant you have. So when you become infected with SARS-CoV-2 and you test positive for it, and if you go get tested, and you tested positive for it, they can sequence your results. Because remember, they've mapped the genome out of these, you know what I'm saying? They know what these variants look like. Because they sequenced it. Now that they sequenced it, they're able to handle business and tell people what has what. So through PCR testing, and they determine the outcome of that. That's how you know. You know what I'm saying? But I don't know what my boy and Bias got going on over there, man. You know what I'm saying? I go over there and hear sisters cussing each other out, man. It's got me worrying about it, man. Like,
This is what it come to. This is what it come to.
Tell him, Tip.
In the fire, I reside like the money do.
That's all. I just wanted y'all to hear the first bar. You know what I'm saying? On biases, this is what it comes to. And it fucked up. I should do all that hollering and cussing. I have 100 people in here. Matter of fact, you know what I'm saying? Yeah, and you seen Unc in the chat over there, but he gonna be like, man, send the link. Nah, I ain't sending you nothing. Go on over there when I'm biased. Pure, you was over there too, gassing it up. We seen you. We read your comment. You and Zane. You and Zane, man. Over there instigating. Y'all don't even know what's going on. There was a lot of noise, though. And this is what it come to. We got a pseudo. I know 2 people that just got COVID. Vax, 1 not vax. I think I want to get the book. Yeah. Oh, to your comments from earlier. Go on and get your booster shot. I ain't gonna tell you not to get it. I ain't telling nobody not to get the booster shot. I ain't doing none of that. I highly recommend people to get the booster shot if you feel the need to get a booster shot. I ain't gonna tell you not to. I'm just not getting it as of right now. I'm still waiting on some signs to come back. Then I really want to get into. Yeah, so let's see what they. I'm gonna play it again one time. Look, Pure still over there. He bouncing back and forth. Pure, what they talking about?
You're bragging about your charges.
Don't forget all of that. And more smoke. Don't forget that party.
You were bragging about your charges.
You mean?
What are they? What are they?
Like I said, look it up.
Your charges are.
Look it up.
Your charges
like
are.
I said, look it up, bitch.
Okay.
That's some homework for you.
Got you.
That's some homework for you.
Amber got charges. Man, we is supposed to be using these platforms to educate our people, man, and get them in the right direction. I mean, I know I've been a part of the ignorance at times too, but at least I'm arguing over information and I ain't arguing over no god dang it identity. I thought we was beyond the identity crisis. But on the better news, man, you know what I'm saying? I would let y'all go on over there and utilize that. So look, it says, hi, Science Sean. Due to copyright match, your stream was interrupted. The lab grown. Oh, okay. Yeah, I already know the stream was interrupted. Why you just send me that? But we back now. We back better than ever. My views didn't went up now. I done called out Pure Black. He on his cell phone and laptop trying to listen to who going to a wannabe Indian and an African American sister with charges. He said I balance ratchetness and scholarship. Yeah, but when I be arguing, though, tell the truth. When I be arguing, it be over the information. For real, I don't be trying to get into the other stuff. But when people when Reggie try to little boy you and all that, I ain't going for none of that. I learned how to mute the noise. Like if you stay away from certain people, you can mute the noise, man, and then it don't even affect you because you ain't paying attention to it. They sent me a link to check out my FICO score. What y'all think my credit score is? What y'all think my credit score is? Somebody put my credit score in the chat. What you think it is? And by the way, it's somebody that's listening right now that has an attitude, a real bad attitude at the present moment. And I know you're listening. So, I just want to let you know that it's kind of monkey that you have an attitude right now, but I know you're listening, you tuned in. And I don't know what I could have done to piss you off so much. You know what I mean? You supported the channel, supported the research team. We appreciate you and all of those things, but I don't know why you're mad. I guess this is what it come to. And they fucked up. Who's at 757? Nah, you wrong. It ain't 757. You wrong. Nice try though. 720 or better. Now you can't do no 720 or better, dude. Guess the score. All this old trying to, you know what I'm saying? If money was on the line and you had to guess, they tell you pick a number between 1 and 10. You can't say 5 and above. Let me see if I can get back to this. I'm gonna find y'all something real quick. I really don't want to take away from Unbiased because I do know all y'all really like to hear the foolishness going on Unbiased channel. Even, especially when you got two sisters just going at each other. It's real easy to just get over there and listen to that. But I do have something that I actually can give y'all temporarily to make y'all evening solid. Do I want to? No, I don't want to mess it up with that. Hold on, y'all. Four hours ago. Okay, let me go on and give it to y'all, and then y'all tell me if y'all can hear. I know they're gonna try to hit us with the copyright, so I ain't even gonna play this. Share this.
Nearly 500,000 new COVID-19 cases were recorded in the U.S. Wednesday. That number from the CDC.
Listen to this very closely. Almost 500,000 COVID cases in one day. We ain't seen that in a long tizzle.
Nearly 500,000 new COVID-19 cases were recorded in the U.S. Wednesday. That number from the CDC is the highest single-day tally since the start of the pandemic. Health officials say getting vaccinated and receiving a booster shot remain the best protection against the highly contagious Omicron variant. As cases continue to climb heading into the new year, the FDA is reportedly close to expanding Pfizer's booster shot eligibility to younger teens and adolescents. Meantime, New Year's Eve celebrations across the country are being scaled back or canceled altogether in response to the virus's rapid spread. CBS's Nancy Chen has all the latest developments from New York City.
It was another record-breaking day here in New York, with nearly 75,000 new cases reported statewide. Hospitalizations are also rising fast here, as more than 1,000 hospitals across the country now say they have a critical staffing shortage.
With final preps underway for the New Year's Eve ball drop in Times Square, COVID cases keep rising to record highs. To fight the surge, incoming New York City Mayor Eric Adams said he'll keep in place a first-in-the-country vaccine mandate for private sector workers.
Our focus is vaccine and testing, vaccine and testing, vaccine and testing.
City services are feeling the strain. Several subway lines have suspended operations because of staffing shortages. The fire department says 30% of its EMS personnel is out sick, and 21% of NYPD officers are out too. In Cincinnati, the mayor declared a state of emergency after more than 24 firefighters came down with COVID, with those remaining forced to work overtime.
Nobody can recall a time in recent history within the last almost 50 years where firefighters have been forced to stay beyond their normal hours.
Nationwide, COVID cases are rising so fast, it's estimated more than 3 Americans are testing positive every second. 15 states, plus D.C. and Puerto Rico, are averaging more daily cases than ever. Florida shattered its state record that was set just yesterday.
Where are we right now in Omicron's surge nationally?
We are at the very beginning, unfortunately, and likely have at least 4 to 8 weeks before we're going to see it rise and then begin to fall again. And during that time, we are going to see COVID activity in this country like we haven't seen since the beginning of the pandemic.
And as pediatric hospitalizations hit a record high for the year, the FDA is reportedly expected to authorize Pfizer boosters for 12 to 15-year-olds early next week, and boosters for children 5 to 11 with compromised immunity. Johnson & Johnson released data showing its booster is effective at reducing hospitalizations against the Omicron variant. Still, no matter their vaccination status, travelers are now being urged to avoid cruises, and new guidance out today from the CDC. More than 5,000 new COVID cases on cruise ships were reported in the last 2 weeks. Omicron's surge has continued to strand those traveling by air. More than 1,000 flights have been canceled today.
We're not going to live forever in a constant crisis, but for the time being, it's going to be a challenge.
Another tool in the fight against COVID is soon on its way. The first of the pills authorized for at-home treatment is expected to go out this week. Elise?
Nancy Chen, thank you. For more on this, we want to bring in Dr. Payal Patel. She is an infectious disease physician at the University of Michigan Medical School. Dr. Patel, thank you so much for joining us. I want to start with news that the FDA is expected to authorize Pfizer's booster for 12 to 15-year-olds. How important is a third dose for this age group?
Yeah, it's a great question. Right now, about half of that age group is vaccinated at all. So that's concerning. Half of those kids haven't gotten a first dose or a second dose. That's really the group that we need to focus in on. And that would be the group that I would really hope parents can hear this news and start getting those first and second doses in. I do think it's good news that they're going to be approved for a booster. And really the part that I'm very optimistic about is that age group of 5 to 11 who have immune deficiencies getting that booster, because we're really trying to protect the highest risk of those kids from being hospitalized or having to come into the hospital.
Speaking of, according to the Department of Health and Human Services, child hospitalizations have nearly doubled in some states. Is Omicron worse for younger patients in terms of severity of symptoms?
At this point, despite us having vaccinations for kids over 5, there remains a huge population of kids who just haven't been vaccinated. And so when you look at the nation and who's been vaccinated and who hasn't been vaccinated, there's just a lot more kids that have been unvaccinated. That's how I would think of it. We don't have evidence yet that Omicron is worse in kids, but I would say I would look at this news and talk to your niece, talk to your nephew, talk to your sister, talk to the kids about getting vaccinated. That's going to be the best way to protect them going into the new year.
So as a parent, if you have a child vaccinated or unvaccinated, when do you advise that you take your child to the hospital if their symptoms are just that severe?
I will say, working right now in the hospital, in the ER, inpatient, it is really chaotic, really busy there. So, a good reason to go to the hospital would be anything like trouble breathing. That would really be concerning for any age. Now, if you're going to the hospital or the ER to get a test, or, something like, you're feeling okay, but you're not sure if you were at a recent birthday party, you may have been exposed. Those are not good reasons right now to come to the ER. And I know here in Michigan, we've actually had to tell the public, listen, our ERs are pretty full. Please don't come just for something like getting tested. So I would say right now, it would be those concerning symptoms that would bring you to the ER.
Can't even imagine having a child that sick. So a new study out of South Africa found a second dose of Johnson & Johnson's COVID vaccine reduced the risk of Omicron hospitalizations by 85%. The CDC recently endorsed Pfizer's and Moderna's boosters over Johnson & Johnson's because of concerns of a rare blood clotting issue. Do you think this will ease fears people may have with Johnson & Johnson shot?
Yeah, and I think this is good news. We are kind of waiting for more data that's going to come out with the new Omicron variant. And this is the latest study in healthcare workers. So these are folks who are going in, working every day, possibly working with COVID patients, possibly being exposed to COVID at home as well. So it's really a real world study, and it's really up to date. I think this is good news for folks who got the first Johnson & Johnson and are thinking about getting a second. We'll have to wait for the final data to come out, but I do take this as hopeful news.
How long do you think it will be before we start to see new infections involving the Omicron variant level off and eventually decline?
Yeah, I do think that it's going to be at least through January that we're going to be seeing what we're seeing now. I think we're only in the early part of the wave. I would remind people, though, that we're in a very different way than we were 2 years ago, when we didn't have a vaccine. And so many people that we knew, if they got infected, were going to end up in the hospital, were going to end up either having a horrible time with the infection or dying. Now, if we think about folks who are vaccinated, ourselves, our family, they're often having a mild infection. And I would say that is really good news, showing that the vaccines do work.
We are definitely in a different state. Speaking of, a lot of people are getting ready to celebrate New Year's Eve Friday. What is your advice to those who plan on attending larger indoor gatherings where the vaccination status of everyone may be unknown?
Oh man, I would say if you're a risk-averse personality, right? This is not the time to play, a battle of risks. You yourself might be honest about taking a test and it being positive or negative, but can you say that for the thousands of other people that are out? So I would really think again, are you someone who potentially could get very sick? Are you older? Do you have any immune deficiencies? Are you unvaccinated? I would take a gut check before you do something this New Year's Eve to ensure that you have many more times in the future that you can get together with those that you love.
Definitely good advice and something to think about. Dr. Payal Patel, thank you.
Thank you.
Tonight, new COVID cases.
They're running that up. Everybody talking about them almost 500,000. Yeah, the news is chomping at the bit over that information, y'all. It's crazy. What y'all think about that, man? Because we've been hitting like 80, 7. We was like at 60, 50, then went down to 50, then to 70, then about 80, and then that joker went up to, you know what I'm saying? Then it went up to 100, then like 120, 200. It went to 300. Now it's almost 500,000. That mug climbed up that much. That's how many people got infected in one day. One day. Craziness. We over here arguing about identity. We got people crying, damn near losing their lives. Losing their lives, man. Okay, I'm telling y'all, this is what it comes to. Mongo over there on Unbiased. That he gonna inbox me, watch. Hey, look, man, I'm gonna shut it down, man. Let y'all get with the foolishness, you know what I'm saying? I appreciate everybody tuning in, man. Y'all go and enjoy the noise, man. Send everybody a happy thought on behalf of me, man. Tell them I said peace.